All right. Uh, Good evening or good morning, everyone, depending on where you are based. My name is Anton Shiriaev. I am very excited to be here. It's an honor to present at the Science Circle, and I'm very excited to explore this new format because for me it's a completely new thing. Here's the outline of my presentation. I will briefly introduce myself. I will introduce the problem of selenium deficiency, which basically was the reason why we started doing this research. Then I explain a little bit of theory about selenium biofortification, about selenium functions in plants, and then I'll talk about our discovery of selenium nanoparticles. I'll show some very few selected results we had over the last 6 years, and then I'll have some quasi-theological or philosophical speculation about why I think science is somehow a divine phenomenon. Yeah, so about my background. I started originally I'm from Moscow, Russia, and I started my bachelor in 2012 at Russian State Agrarian University. My specialization was plant genetics and plant breeding at the Faculty of Agronomy. At the same place I did my master. But during my master degree, I moved to Hungary, to St. István University, to study agricultural biotechnology for 1 year. I was very excited about living in Europe, about having different educational approach, different lifestyle. So when it was time to think about PhD, I was exploring opportunities in Europe, and that's how I came to Pisa, Italy, where I am still based up to now. I did my PhD at St. Anna School of Advanced Studies. My curriculum called Agrobiosciences, but generally it was about agricultural biotechnology, I would say, mostly. Then during my PhD, I went for a 4 months long period abroad in France, where I was working with transcriptomics, encrypting the transcriptome of our tomatoes. And after concluding my PhD, I moved as a postdoc to Italian National Research Council, where I'm still serving as a postdoc. And finally, now I am visiting with a very short visit University of Verona, which is on the north of Italy, but I'm here only for 1 week. But anyway, greeting you all from a beautiful northern Italian town called Verona. So first I wanted to explain the difference between two concepts which for me seem to be very similar at the very beginning. One concept is starvation and another one is malnutrition. Often they're mistaken, but the key difference is that starvation is basically a lack of calories in the human diet. And malnutrition is the lack of minerals, nutrients, bioactive compounds, vitamins. And surprisingly, nowadays malnutrition is more severe problem globally than starvation. So there are more people having lack of essential vitamins and minerals than the number of people who do not have enough calories in their diet. And traditionally, calories are mostly obtained from staple crops, and what we call staple crops are so the main crop providing food for people. For example, in Asia it's rice, in Europe it would be more wheat, somewhere it's potato, in Central America I believe it's more corn, and vitamins are mainly received from fruit and vegetables. So having a balanced diet and protecting ourselves from malnutrition, we should have also enough fruit and vegetables on our table. And one of the examples of malnutrition is the selenium deficiency. So, for example, in China, there was a study aimed to correlate selenium content in soil with certain diseases which are supposed to be correlated with a severe selenium deficiency. And indeed, as you could see on this slide, on the left part, we could see the concentration of selenium on the soil, and on the right side, we could see the spread of 3 diseases affiliated with selenium deficiency. In Europe, selenium content is also not very high, selenium content in the soil. And even though nowadays we are eating pretty geographically diverse, let's say, products, and we might think that if we are consuming, I don't know, apples from Italy, potato from Central America, grapes from I don't know, France, and if selenium content in the soil is different in all these places, we could assume that, yeah, maybe it's not so important for us to take care about selenium level. However, my personal experience is that I am selenium deficient, and I'm living in Europe for the last 6 years. Moreover, I've been consuming some selenium biofortified products over the last 6 years, and I'm still selenium deficient. But also experimenting on myself, I see that this problem is it's here. So it's not just somewhere in the areas where people consuming only local food, but it also can be valid for many of us. But why selenium is important for humans? Well, selenium takes parts in some enzymes, some hormones, for example, thyroid hormones, and it's surprisingly important for many different functions in our body. So it somehow contributes to proper functioning of immune system, of brain function, of reproductive function, especially for males. And also it was somehow observed to be able to support, it has some anti-mutagenic effect as well. So those people who are selenium deficient, they are trying to find some external sources for balancing their selenium level. And one of the sources are natural selenium accumulators. So some plants tend to accumulate more selenium from environments than others. And for example, 2 most common edible selenium hyperaccumulators are Brazil nuts and garlic. So one way to balance our selenium disbalance is consuming garlic and Brazil nuts, but not all people love these products. So some prefer to have food supplements with selenium. However, not all food supplements containing selenium are the same, because our body is not capable to transform mineral selenium into organic forms, which we basically need to construct those important hormones. And one of the most common food supplements containing selenium is the extract of spirulina enriched with selenium. However, spirulina is basically a very simple organism, and it's not efficient enough to transform mineral selenium into organic forms. So basically what we are consuming, taking this kind of food supplements, is spirulina enriched with mineral selenium, which is not absorbable by our bodies. However, if we will consume selenium in organic forms, so for example, selenium methionine, it might be more efficient. Well, what about selenium in the environment? So initially selenium is stored somewhere deep down in the earth crust. It's taken up by volcanic activity, also by groundwater. It's spread on the surface with water, and also sometimes during mining activities. For example, in Chile, soil is very rich with selenium, mainly due to the very active mining activity. However, as I said before, the main form of selenium in the soil is mineral form. So basically selenate or selenite, those are two main forms of selenium. And if you could see this scheme, plants are here very important because they are the ones which are transforming mineral selenium into organic molecules, which then can be consumed by animals or also by humans. So we assume that plant biomass is the key source of bioavailable selenium in our diet. And in Europe, already for several years, if you go to a supermarket, you could find there selenium-enriched products. For example, this company produces selenium-enriched potato, selenium-enriched onion, and carrot. And I think it's very interesting product, not only from the nutritional perspective, but also it's somehow raising awareness about malnutrition. It's raising awareness about technology. It's raising awareness about our health from another perspective. However, there were performed some studies in China on selenium-enriched rice, and they found that after 5 minutes at the boiling temperature, organic selenium oxidizes back to inorganic forms. So unless we would compromise on taste and texture, and we would agree to eat raw potato, it might be not the best way to balance our selenium deficiency consuming selenium-enriched cooked potato. Probably it's easier to obtain enough selenium from fresh products. And thinking about fresh products, we start thinking about perishable foods and leafy vegetables. So before we spoke about selenium functions in our diet, in our bodies, but what about selenium functions in plants? If we use them as a carriers of our dietary selenium, what it does inside plants? Well, surprisingly, selenium it's not essential for plants. So if you will cultivate any plant on selenium deficient substrate, there will be no difference. There will be no alteration of important functions. This is more precise explanation. However, there are some studies where selenium was used as a treatment, also to challenge, to test what effects it makes on plant physiology. And there are some studies showing that it affects biochemical composition of plants. It somehow positively impacts pathogen resistance, and this can be explained by the direct effect of selenium, which can be toxic, and maybe it interacts directly with pathogens, and the selenium-enriched plants are somehow containing, well, basically poison for pathogens. Another interesting observation is that selenium was capable to alterate growth, photosynthesis, and also primary and secondary metabolism. So it's very interesting that evolutionary selenium, so plants grew up as a kingdom without considering selenium as something important. But now somehow we see that it does something. It does something with plants. And one important thing is that some studies are also showing that selenium treatment is capable to postpone fruit ripening in tomatoes. I'm very patient about tomatoes. I studied them for most of my academic career, and one of the main problem in producing tomatoes is that they are so fragile, is that they are so susceptible for diseases, and that sometimes farmers are losing up to 40% of their initial harvest. So from the moment we harvest to the moment we see the product on the shelf, well, 40% is already lost somewhere on its way. So every time when there is an opportunity to develop a technology or to contribute in developing technology potentially capable to decrease post-harvest losses in tomatoes, it's usually a very interesting challenge for those working with tomatoes as the model objects. What about chemistry of selenium inside the plant? So, as I said, selenium is not essential for plants. So plants even did not develop selenium transporters. And the way selenium travels inside plant is that they are taking advantage of sulfur transporters, because chemically selenium is very similar with sulfur. And the same observation was made by Berzelius, who discovered selenium. So he was first thinking that it's some sort of very strange form of sulfur, but indeed it was a new element, selenium. So what we know about the transport of selenium? It's absorbed by selenium transporters by sulfur transporters in the root. Then it's taken up with a xylem suck. It's transformed into organic forms, selenium cysteine and selenium methionine, in cytoplasm, mainly in leaves, and then it can be transported to fruit, where it's stored as organic molecules, or it can be evaporated in some volatile forms. So another application of plants can be to use them for phytoremediating soils polluted with selenium. So they can absorb it as a mineral selenium, they can transform it into organic forms, and then to emit it as volatile forms in the air, cleaning up the polluted soil. I'm still struggling from time to time to switch the slides. Yep. So what are the strategies to enrich plants with selenium? Traditionally, selenium was used in form of sodium selenate, more rarely as sodium selenite, because selenate is more toxic. It's not so common to find studies working with selenate. So usually it was added to the substrate or to the nutrient solution, and then it was absorbed with roots, well, basically following the mechanism I showed on the previous slide. However, at high concentrations, selenium can be toxic, and with the current trend for sustainability and also sustainability in agriculture, it's important to think what we do with the selenium solution after cultivating our plants. So we cannot just spoil fields with a lot of mineral selenium. So what could be the other options how we can fortify plants with selenium? One of the options can be the use of nanoparticles. And nanoparticles are basically more developed in the field of medicine, where they were discovered to be very precise in delivering drugs to specific sites of our bodies. But the same mechanism can be applied in agriculture. So nanoparticles are very tiny. We can use any form of chemical inside of this capsule, and we do not care about chemical form of the substance. And for example, with selenium, we can design nanoparticles containing elementary selenium, so selenium-0, selenium which does not have electric charge, and which is basically not perceptible by plants as a free form, because they need to have some charge to kind of to transport it. And nanoparticles having their own charge can be fortified with a neutral, non-toxic selenium. So even though these nanoparticles can then go to the environment, they will keep inert, they will not interact with any other living organism or plants. They were also found to be more effective in some studies comparing to sodium selenate, and also, as I said before, they can be more sustainable or more safe. Also for those working with the selenium, so those operating the treatments. And so during my PhD, we studied a series of experiments where we were using L-cysteine-based, chemically produced selenium nanoparticles. They were about 100 nanometers in diameter. We were spraying them on plants at the very, very early stages of fruit development, so between flowering and the immature green stage of fruit development. When you hear the pause, it means that I'm struggling to switch slides, so I appreciate your patience. That's what we were doing in the greenhouse. So we had a pretty large set of plants which we were spraying with selenium nanoparticles at different concentrations. We were also spraying them with another set of plants with sodium selenate to then make a comparison of which technique is more effective. And then after randomizing them, we were just following the usual flow of the experiment. Here are the first results. So we saw that mainly selenium was accumulated in the vegetative parts of plants, so in leaves and in stems. And in fruit we had pretty low concentration of selenium. However, it was there, which means that it was transported from leaves to fruit. We also saw that sodium selenate was more effective than selenium nanoparticles. So plants accumulated more selenium in it than in the form of nanoparticles. nanoparticles. Also, we saw that over the time, selenium has moved from vegetative parts of plants to fruit. So it was the amount of selenium was dropping in the leaves, and it was raising in the fruit. Seeing this very low concentrations of selenium inside the fruit, we were thinking, well, what could be the other options? How we can have more selenium in the fruit? And we had these two very interesting experiments where we were trying to biofortify already harvested fruit, which did not reach their final ripening stages, and we were fortifying them with selenium solution after passive immersion in the solution, but also after applying negative pressure with a vacuum pump. And we saw that, yeah, with these techniques we had a massive accumulation of selenium inside the fruit. However, we could already guess that the time was too short to transform this selenium into valuable organic forms. So we kind of suspended these experiments, focusing more on treating the whole plant. These are our beautiful experimentals. Huge amount of samplings, a huge amount of analysis to understand what selenium does with this fruit after it gets in. So we performed a massive amount of targeted and untargeted analysis to study quality and composition of this fruit. So I had a huge opportunity to explore a vast diversity of different techniques, starting from gas chromatography to GCMS to nuclear magnetic resonance to liquid chromatography, mass spectrometry to atomic absorption. So a lot of fascinating and very fun techniques which allowed us to harvest very interesting data. So one of the first pieces of the data I want to show is the effect of selenium on volatiles, which we were capturing with gas chromatography, mass spectrometry. Yep. So please don't worry about having very tiny names of the compounds. I wanted just to show the volume of the data we had, the amount of compounds we were able to measure, and to show that there is pretty good clustering of some compounds with ripening stage, but also with selenium content. So the selenium content is growing from up to down, and the ripening is moving from left to right. And based on this analysis, we found that volatile profile was impacted, and especially aldehydes, terpenoids, alcohols. We also saw that flavonoids, measured with nuclear magnetic resonance, with another technique, not with nuclear magnetic resonance, they were also changed, and their composition was improved. And it's good news because flavonoids and polyphenols are also desirable in our diet. They have antioxidant properties, and so we can already conclude that that selenium can improve the nutritional quality of tomatoes directly by increasing the level of selenium itself, but also indirectly by pushing forward the production of bioactive compounds, for example, flavonoids and polyphenols. Also, interestingly enough, some of the volatiles, and this is the data from another experiment, one of my first attempts to work with volatile organic compounds. So we saw that some of the volatiles which were found to be positively associated with consumer liking were increased by selenium. So we can also assume that selenium improves taste, improves the aroma of tomato, which is pleasant. But what about ripening? So here I will introduce a little piece of theory, which is important to move further. So basically, there are two kinds of fruit, depending on their respiration pattern and their interaction with ethylene. There are climacteric and non-climacteric fruits. Non-climacteric fruits are having very scalar respiration rate for both emitting CO2 and ethylene. So they don't have any, let's say, anomalies in their respiration path, while climacteric fruit are having this dramatic increase of CO2 production, of respiration, and of ethylene release at the last phases of their fruit maturity. And good examples of non-climacteric fruit is strawberry, and tomato is assumed to be one of the model objects for studying physiology of climacteric fruit. But why we have this hill on the graph? Why we have this dramatic increase of ethylene production in climacteric fruit? Well, somehow ripening of climacteric fruit is ethylene dependent, and climacteric fruit are able not only produce ethylene themselves, but they are also capable to have ethylene autocatalysis. So if you treat them with external ethylene, they start ripening faster. That's why it might be wise not to store bananas close to avocados if you don't want them to ripen faster together, because both of them are producing a pretty good amount of ethylene, which can trigger the production of autocatalytic ethylene in one another. About ethylene synthesis, so it's pretty complex process, and so I want to introduce this wonderful scientist, Professor Shanfa Yang, who discovered the methionine cycle, or Yang cycle, as it's also called. Is basically a recycling of the part of the molecule which then contributes to the formation of ethylene molecule. And so there are many steps in ethylene production. Each step is controlled by certain genes, and there are massive amount of genes involved in ethylene synthesis. And so people working with ripening, they not only interested in seeing physiology of ethylene, but they're also interested in selenium regulation. So they're often interested in the regulation of genes involved in production of ethylene and in sensing of perception of ethylene in climacteric fruit. So then I wanted to briefly mention the central dogma, which states that the flow of information goes from DNA through RNA to protein, and there are three key processes, which are replication, transcription, and translation. And now I will introduce some data which we obtained after encrypting the transcriptome of tomato biofortified with selenium. So what we did, we read all the RNA contained in tomato fruit treated with selenium, and we compared the expression of this gene, of these genes, with expression of genes in tomatoes which were not fortified with selenium, and eventually we had some set of differentially expressed genes based on which we could say we could list the genes function of which was impacted by selenium treatment. And what we saw is that there is a massive group of genes involved in ethylene signal transduction and consecutively in ethylene synthesis in tomato fruit. Unfortunately, in the experiment where I was working, we haven't seen any changes in ethylene production, so we were constantly measuring the release of ethylene in the fruit. However, we haven't seen it in this experiment. However, my colleagues, before I joined the group, they did some analysis of ethylene production, and they saw in another trial that they had 2 days of difference in the appearance of ethylene peak. And it's basically, it means that selenium could postpone ripening. But since we cannot replicate it, and we cannot see this in the samples on which we had this transcriptomic data, it's very difficult to discuss. But we're very happy about both physiological and transcriptomic results of selenium effect on ethylene. Then about flavonoids. Yeah, it's always nice to see some results which can be translated, which can be applied to improve our lives. But what about the basic research? What about the explaining why certain things are happening? So why and how selenium impacts flavonoid biosynthesis. So what we saw in our RNA-seq data is that selenium upregulated 3 genes involved in coumarate pathway with the final project of naringenin. And again, naringenin is a flavonoid with antioxidant capacities, so we are happy to have more naringenin. And also so when we analyzed flavonoids only in mature green stage, we saw indeed that coumarate level was decreased and naringenin level was increased. And this data matches very well with our transcriptomic data. So basically we have more source, and we have less source because these genes are working very well, they're working fast, and they are metabolizing coumarate into naringenin, and we have more naringenin at the very end. Switching the slide. Then also we saw some effect on amino acids, and the synthesis of amino acid is basically, amino acids is the primary metabolism, so very important part of physiology. It's very interesting to see that not essential element can do something with primary metabolism. And one of the genes which was downregulated is the trianyl tRNA synthetase. And what it does, it basically delivers threonine molecules to tRNA, which then ships this amino acid to their future, to construction of proteins, following the central dogma which we repeated before. And so we can assume that maybe if the expression of this gene is downregulated, it means that these guys are working pretty slow, so we should see an increase of threonine content. And that's exactly what we saw when we analyzed the content of threonine in tomato fruit. So now I'm coming to the conclusions. First of all, we designed 3 strategies which can be used to produce selenium-enriched tomato fruit. And these tomato fruit have enough selenium to supplement our diet with beneficial organic selenium molecules. At the same time, selenium, which used to be an essential poison when it was just discovered, so the level of selenium in this fruit is not reaching the toxic level, and using tomato fruit, for example, as a carrier of this selenium can provide more security that we will not accumulate a toxic amount of selenium, because also plants have their own ways to regulate selenium uptake, and they will not accumulate it more than they can. Then we could say that the effect of selenium on ripening was positive, but we still need to work on it, and potentially selenium can help us to decrease post-harvest losses in tomato production. Also, we saw some positive changes in metabolome, which can indicate better taste and better aroma profile. And some little practical remarks. So selenium deficiency can cause some health disorders, and to obtain recommended daily uptake, it's enough to eat 2 Brazil nuts per day. If you don't like Brazil nuts, eat garlic. If you don't like garlic, please keep an eye on our research, and hopefully we'll have some selenium fortified tomato fruit on the market. Who knows? Then it's also important to remember that effectiveness of biofortified crops and food supplements depends on the form of the element inside. Heat is capable to destroy bioavailable organic selenium molecules to its inorganic forms, and selenium biofortified products becomes basically not very helpful. Tomatoes are climacteric fruit, and ethylene is very important for their ripening. It's very important for their physiology. So when you store tomatoes, also depending on your aim, if you want them to ripen faster, put them close to bananas. If you don't want them to ripen too fast, keep them away from another climacteric fruit. Well, and now I have 2 minutes to say why we played GAT 3 times. So first, there are no selenium transporters in plants. And I think it's very interesting to think that we took advantage of the nature which made a mistake mixing up sulfur molecules with selenium molecules. And like cuckoo birds which are laying eggs in the nests of other birds, well, I feel proud that we had this play with the nature, and I feel proud that this play worked. Next, we wanted to use elementary selenium, but elementary selenium didn't have a charge, so it cannot be accumulated using, let's say, traditional transport mechanisms inside plants. But we used nanoparticles, which were protecting this tiny imposter, little elementary selenium sealed inside the organic shell. And it came to me this metaphor that it reminds this ancient Greek myth about the Trojan horse, which was used to conquer the town um, yeah, with some armed guys sitting inside the wooden figure of the horse. And lastly, uh, I don't want to open the debate about GMO, um, but I really like to speak with people who are concerned about GMO. I really like to talk with them and to ask them what is their opinion about changing the function of the gene without modifying the sequence of the genome. And usually this conversation ending up very interestingly. Um, and that's exactly what we actually did. We alterated the function of genes without changing the genome, and it's very I think it's very soft way to start speaking with GMO denials. Uh, Even though the changes of the genes, of the gene expression we observed did not cause to um, big changes in physiology. So we did not observe uh, a massive decrease of ethylene production or massive delay of ethylene peak, but we were very close. And I think this sense of being so close, it's very pleasant. And it reminds me this this art piece. If I will manage to, yeah. So this is one of my favorite artworks, and the whole beauty of this artwork, which was noticed by one of the Russian poets, Joseph Brodsky, is in this fragment on the left. So the mother supporting the child, but at the same time she's not touching his foot. It's very, very close, but it's not touching. So we know that child is protected, but we also see that there is a little the little distance between child and the mother's hand. And this Russian poet said that the entire beauty of the universe is sealed in this 1 millimeter of the distance between mother's hand and the child. I want to thank Stephen for inviting me to have this talk. I want to thank my supervisors, Professor Pietro Tonutti, Dr. Beatrice Pezzarossa, and other members of our research group. And thank you for letting me share. I would be happy to address your questions. Also, please email me, search for me in LinkedIn. I would be happy to also to have some individual talks. Thank you.